the first thing I want to do is ask you guys how last week went, since that was, we haven't done that kind of thing for a while.
Thank you.
What,
Yeah.
I mean, were they frustrated by trying to do the bioinformatics and the science and stuff like that?
It's too bad.
It wasn't too bad.
I thought it was more of a challenge than like the usual ones, but it was like people, yeah, they
I don't
weren't
think they
too
were
frustrated.
time though. I think
They had enough time to do it. So are there changes that you removed in terms of what we did? Any suggestions about
I
that?
don't think so. It was a perfect length too.
Okay.
They all finished in time.
Okay, so hopefully, so then the next question is, did they get a good idea of databases, genome databases from it? Or was it just like plug and chug and I don't care?
Might be like a little too guided to be, maybe like, could be a little more, I don't know, less like, like grabbing your hand and walking you through it. And so, I don't know. But I thought, like, it's a complicated interface, you know, BLAST, so it's like,
It is complicated, yeah.
And like all the parameters are not super intuitive. It's too important maybe.
yeah, it's
All the-
not,
I'm
not
sorry,
very
all the way
intuitive.
down?
All like the results you get from a search, they're
Yeah.
not intuitive.
They are, yeah, when you get that feed, that,
Yeah.
it's hard to figure out that
Right.
first band actually matches the sequence that's
Mhm,
below,
yeah.
that kind of
And
stuff.
like what identity and what max score mean.
Yeah.
Like e-value. Like, work through it, yeah.
So did the worksheet help with that or not?
I think working through it made it a little clearer, but I'm not sure.
So this is what I'd like you guys to do then, if you could, is go back over it and see, like Duncan, where do you think they could have done more independent work? Where could we explain it a little bit less or more? What would have helped? And then just give them those suggestions. Not today, but maybe by next week or so. Well, actually, next week we
Okay.
have the exam.
Oh yeah.
So we won't be working, we won't be meeting, okay? Do you-
Yeah.
Wouldn't it be a matter of expressivity? You know, like if you were missing one, or do you think you only need like a minimal amount of functioning enzyme to like have that effect? Like if you have P53, if you only
That's
had
what I was thinking. Because you can still not have cancer with one functional P53.
so you might only need one functional version of the gene then. Do you know anything about that?
So this is P53, it's a tumor suppressor, right?
Yeah.
Yes.
Right.
So at the
So-
organismal level, is it dominant or recessive?
So, yeah, so what do you think? You guys tell me. So you're
Generally.
Well, would they? I guess generally recessive still, right?
at the organism level, yeah. So why would you say that?
Well, then do you only need like one functional version of a tumor suppressor gene to not contribute to a cancer phenotype?
So do you only need one to not contribute? Is that what you're saying?
Yeah.
Sorry,
Do you like
I'm
so?
listening to a negative.
Sorry. Do you only? Is it alright to only have one version of?
Right, so one wild type copy versus one mutant copy,
Yeah.
right? So can you inherit tumor suppressor genes?
Yes.
That's the first thing, yeah, you can, right? Okay. And so why, so then what is the problem if you have a mutation in one of the tumor suppressor genes that you have?
So couldn't the environment mutate your healthy one?
Awesome heterogeneity.
Yeah. And it's getting back to what you're asking about too, which is the expressivity. So yeah, so if you get a hit in the other gene, then you have a total loss of heterozygosity, right? And now, what the tumor suppressor genes, what do they do? You guys know,
What?
one's the accelerator and one's
Well, they're
the
just
brakes. Right, and so now you have no brakes in your car, right? Which means what? Means that you're gonna do what?
I mean, like you're gonna develop cancer, but if
Yeah.
you are heterozygous for tumor suppressor, you don't.
So you're,
Cancer,
it's not dominant at that point, but at the organismal level, it turns out it is thought to be dominant because you have a much more higher incidence of cancer.
So because of the higher incidence.
So it's a funny thing to think about, and the thing we should ask is, should we ask them that? Okay.
I think it's a good question.
I think it's a good question too.
Yeah.
So one functional copy of the gene isn't sufficient.
It is. It's fine. So
It's not saying the LNAM minimum, but the increased incidence is why they call it dominant. Because it's so much higher
So-
when you get cancer, because you only have one copy, the other one is mutated.
So it's not dominant in the sense of traditional recessive dominance that we think of.
Yeah.
It's dominant in the fact that
Much more.
How does just seeing like a green fluorescence let you know that they're overexpressed or underexpressed?
Okay.
Is this the intensity?
So let's draw that on the board. Do we have a green marker? Somebody else draw, because no one's got it there a couple of times. One blue, okay, so you do zigzag, or, do a dashed and then a straight line for the other. Will, are you volunteering?
Yeah, I'm volunteering, because I'm still a bit confused myself.
Okay, good, so and then the next question is this a good way to ask this? Because she's doing non-proliferates. And so think about that and see after we look at